Alright guys, this is a review for the second week review as opposed to 3 weeks. We're going to cover the cell cycle, DNA replication, mitosis, and meiosis. Alright, what you guys are going to do, I gave you a paper in the back of your paper review. There is this for you to fill out. Alright, you're going to fill it out completely, so you're going to have to take the time to really go through this. I'm going to break each section into parts. We're going to go ahead and talk about DNA structure and function. And then we're going to go ahead and talk about the cell cycle. Remember, the cell cycle is a series of steps the cell goes through in order to replicate. So we're going to go through the different steps as to G1, S, G2, and then the M phase. And then we'll go into meiosis, which is another form of cell division, but that's for your sex cells, for your gametic cells. So let's go ahead and get started. So we'll go ahead and start off with this basic structure and function of DNA. DNA is a double-stranded nucleic acid, alright? It's double-stranded and it's made up of monomers called nucleotides. These nucleotides are made up of a phosphate, a 5-carbon sugar, and a nitrogenous base, nitrogen base, or a base. Just know that the phosphate and sugar are the backbone of DNA, and the nitrogen bases are what holds the genetic code, alright? Are what holds the genetic code. There's 4 types of nitrogen bases, and I got my little homeboy, my little homie here, called GCAT. GCAT here tells you 4 things. How these pair up, how the nitrogenous bases pair up. G goes with C, A goes with T. So guanine goes with cytosine, adenine goes with thymine, and it also tells you the 4 nitrogenous bases. Alright? So GCAT's very essential in understanding the structure of DNA. So here you have your ladder, and the double helix is a twisted ladder. Alright, a twisted ladder. So just imagine a ladder being twisted, and that's how you would get your double helix. Here's a simplified version of DNA with a phosphate sugar backbone. So here's a phosphate, the sugar, and the nitrogenous bases based on Chargaff's rule or the base pair rule. A always matches up with T. G matches up with C. That's why I'm saying that GCAT will help you understand all that, okay? And what holds the 2 sides of DNA together, the 2 sides of DNA together here are held by nitrogen bonds. So here's the DNA structure, all right? The bases are arranged in triplets or 3 letters at a time. So you're going to see, or you've seen, where we ask you for the complementary strands. That means find the strand that matches up with all this up here. Alright, that matches with this. So A goes with T, G goes with C, G goes with C, and so forth. So this is what you're going to be looking for when we ask for the complementary strand. Alright, especially in your review. That's the complementary strand. It's going to be 3 letters at a time, known as triplets, and also codons. Also know, we spoke about the key scientists that were responsible for the structure of DNA. The first one was Rosalind Franklin. Rosalind Franklin studied DNA using X-rays, so she was able to produce this and determine that there was a 3-dimensional structure of DNA. But then these 2 guys, Watson and Crick, back in 1953, made the first chemical structure model of DNA, as you can see them here. Alright, Rosalind Franklin took the X-ray. These 2 guys used her information to develop the 3-dimensional structure, chemical structure of DNA. This is part of that review sheet I gave you for the back. So the top part of it's going to show this. Just know that a double helix is your DNA. That coils up together to form chromatin. Chromatin then forms chromosomes. Chromosomes are made up of sister chromatids. Alright, sister chromatids. Alright, so let's review that. Here you have your chromosome made up of sister chromatids. These chromosomes are made up of chromatin, which are supercoils. And those chromatins are made up of the double helix, all right? Made up of the double helix DNA that's tightly wound. Also know that chromosomes are made up of DNA, and chromosomes are also made up of genes. Genes are segments of DNA, pieces of DNA that code for your traits. So the code is held within these nitrogen bases. Remember, all living things have the same 4 letters. But what sets it apart, it's the way these letters are set up, the sequence of letters. The arrangement of the letters is what sets us apart, okay? The way the genes are set up, the way the nitrogenous bases are set up. That's what sets us apart, okay? So here we go, the cell cycle. The cell cycle is made up of a series of steps that cell goes through in order for a cell to replicate itself. So here you start off with G1, okay? Then it goes to the S phase, G2, and then the M phase. Just know that your somatic cells are your body cells. They grow, they replace, they repair, and that's an asexual reproduction. So in G1, you have normal cell growth. You get your cell and it doubles in size. Still has 46 chromosomes. And let's talk about your 2N. Your 2N is your diploid number. Diploid number is, you have 23 pairs. So 23 pairs times 2 is 46 chromosomes. That's your 2N number. So then, that's G1. S is the synthesis phase. This is where DNA replication occurs, which I'm going to talk about in a little bit. DNA replication has 3 steps. You have your double helix. The double helix unzips with the help of DNA helicase. Then you start forming 2 new strands. They're semi-conservative. What that means, you have 1 old strand and 1 new strand. So you get 2 identical strands, all right? At the end of the S phase, instead of having 46 chromosomes, now you have 92 because you duplicated, you replicated your DNA. So there's checkpoints here. So here, if there's something wrong with the cell would go through what they call apoptosis, where it ends up shutting itself down and destroying itself. Here, there's another cell checkpoint, where if something goes wrong, if there's letters missing or the letters got mixed up, same thing happens. It repairs itself before it moves on to the next phase, which is the G2 phase. G2 phase is the final check for mistakes. It fixes, sends it back, or it kills the cell. But these 3 phases of the cell cycle are the longest phase, make up the longest phase of the cell cycle, which is interphase, okay? Then we go into the M phase, which I'm going to go into in a little bit. Just know that the M phase is where actual cell division occurs. So here are models of the cell cycle, and they all start off with G1, S, and G2. That's all part of interphase, as you can see here. And then you have the mitotic phase where actual cell division occurs. All right, some models show you G0 where the cell is at rest. G1, all right, this is where normal cell growth and development. Then you go to the S phase where DNA replication occurs, the synthesis phase. Then you go to G2 where the cell double checks, all right, for the duplicated chromosomes for any errors and makes any necessary repairs. And then it goes into the M phase, another model of the cell cycle. So here's DNA replication. Just know that it's 3 steps. You have 2 strands of DNA. The process is semi-conservative. All right. The DNA unzips with the help of DNA helicase. The 2 new strands form with the help of DNA polymerase, which are 2 enzymes that help out during this process. These free-floating nucleotides start attaching to the exposed strands, and you end up with 2 exact copies. But once again, it's semi-conservative. We have 1 old copy and 1 new copy. All right. Just note that there's checkpoints at every phase, and if something goes wrong, either the cell fixes the mistake or it kills itself. Now the M phase. The M phase is known as the mitotic phase. All right, the mitotic phase. You have prophase, metaphase, anaphase, telophase, and at the very end, cytokinesis. Okay. So the way this works, you have G2 where it checks for the final mistakes. Then it goes into the M phase. In the M phase, the cell prepares for cell division. Okay. And then in the metaphase, which is the second step of mitosis, the chromosomes align in the middle. So meta-middle, all right? Meta-middle. The chromosomes align in the middle. Prophase, the cell prepares for mitosis. Metaphase, the chromosomes line up in the middle. Anaphase, they begin to pull apart, all right? They begin to pull apart. The chromosomes begin to pull apart with the help of the with the help of the spindle fibers, all right? So the sister chromatids begin to move to opposite ends. In a telophase, you start seeing the 2 cells. They haven't exactly split yet. That's not into cytokinesis. That's where the cytoplasm divides and you have the 2 cells. All right, so here's mitosis again. Prophase prepares for cell division. All right. Then you have metaphase where the chromosomes line up in the middle. Then you have anaphase where the spindle fibers begin to separate the chromosomes. Telophase is we start seeing, all right, the indentations right here at the end. We start seeing the 2 cells. So get familiar with these pictures here because you might see those soon, all right? Just know the basics of meiosis. The basic of meiosis is that you're trying to produce gametic cells as opposed to mitosis. Mitosis, cell division for body cells, somatic cells. But meiosis is cell division to create gametic cells, your gametes, all right, egg and sperm. So what happens, you start off with a cell that has 46 chromosomes, but then you end up with cells that only have 23 chromosomes. 4 haploid cells. They have half the number of the original cell. All right. The one thing you have to remember about meiosis is the reason we're all different and there's a lot of genetic variation among the species is because during this process, pieces of the chromosomes cross over and you get a whole new variety of genes, as you could see here. All right. You see the process from here to here crossing over, which is also known as recombination. So meiosis, the whole process of meiosis, just to keep it simple, you're forming gametic cells, which are your eggs and your sperm, and you go from a 2N from 46 chromosomes down to half the number for each cell, 4 genetically different cells known as haploid cells, gametic cells. So here we go. The ovaries and the testes, they go through meiosis, they produce the egg and the sperm, each have half the number, a haploid number, half the number, they meet. Now you form the 46 chromosomes, all right? Now you form the 46 chromosomes. So you go from 23 in each gametic cell. Then you finally form a diploid zygote, which has 46 chromosomes, and then that produces us going through the cell cycle. That's how you get from going from one cell to a trillion cells in multicellular organisms. Here's another example. Mitosis. At the start, you have a diploid cell, and you end up with identical cells. In meiosis, you end up with 4 genetically different haploid cells having half the number. Okay, huge difference, both cell divisions. So here's the final product. Okay, the final product, this is what you're going to have to fill out. I went through the review fairly quickly, so it's a little bit longer than what I anticipated, so I hope you use this to study and to complete your paper review, and go back and review this, and it'll help you along the test. Best of luck, guys. I hope you study and enjoy the weekend.